Hello, everyone, and thank you for joining this month's Bench to Bedside webinar series. I'm Nicole Wilson, Program Manager for the United Mitochondrial Disease Foundation. We're so glad you're here. Before we get started, I would like to remind everybody about MitoShare, our worldwide patient-populated registry. This is the ideal place to help capture the overall journey that our patients experience. So please let your patients know about this important registry today. Save the date for MitoMed 2025 in St. Louis, Missouri, June 18th to the 21st. You can learn more at umdfconference.org. We hope we get to see you there. And now we'd like to welcome our moderators for today, Dr. Amel Kara with Massachusetts General and Dr. Philip Yeski with the United Mitochondrial Disease Foundation.
Hi, everyone. Pleasure to be here with you today. We will be getting a series of updates over the next hour. I am actually going to turn it over to the chair of our Scientific and Medical Advisory Board, Dr. Amel Kara, to help moderate the session. Amel?
Good afternoon, everyone, and welcome to this UMDF Bench to Bedside series. We're very excited today to be with our guests who are going to give us an update on the clinical trial landscape in mitochondrial medicine. It's very exciting to see so many programs out there, and today you are going to hear from clinical trials that are underway, clinical trials in preparation, and get some updates on ongoing efforts to come up with a therapeutic strategy for mitochondrial disease. Our first speaker is Dr. Natalie Yevgeohana. She is a PhD scientist in mitochondrial biology and has led Minovia since its founding in 2012. She will provide an update on the clinical development program for Minovia's mitochondrial augmentation technology. Natalie, welcome, and please take it over.
Thank you, Mel. It's a pleasure to be here. Good afternoon, everyone. So I'm going to present the clinical progress with our mitochondrial augmentation technology. So our lead product is MNV201. This is a cell therapy that is autologous, derived from autologous hematopoietic stem cells enriched with allogeneic placenta-derived mitochondria. In the first step, the patient is being mobilized with drugs that mobilize stem cells to the peripheral blood. Then the stem cells are augmented with healthy and functional mitochondria derived from our cryobank. In a 24-hour process, the mitochondria are internalized into the hematopoietic stem cells, and then the cells are IV injected to the patient. Can everyone see the slides? Okay, yeah. So I want to update today about our phase 1b open label single dose clinical study to evaluate the safety and therapeutic effect of MNV201 in pediatric patients with Pearson syndrome. The target population are Pearson patients suffering from a deletion in the mitochondrial DNA genome that is observed in the blood, and also suffer anemia or other cytopenias and blood transfusion dependency. The goal was to target the 5 patients for this clinical study with up to 20 million cells per kilogram body weight. It's an IV injection, and the primary endpoint is, of course, safety. And secondary endpoint, we measure different anemia parameters, hemoglobin levels, blood transfusion dependency, red blood cells levels, and of course the questionnaire that measures how the patient feels and function, which is the IPMDS questionnaire. We are also looking at other parameters, and we will look for blood biomarkers for our mitochondrial content and function, and there are a few times for follow-ups. For the update, so I can tell that so far, 5 patients have been treated with MNV201 with primary mitochondrial DNA deletion syndromes. 2 patients were KSS patients. So you can go forward to this next slide. 2 patients with KSS were dosed under a compassionate use program with the longest follow-up of 10 months now for the first patient. 3 patients were dosed under the clinical trial, the phase 1b, with Pearson syndrome. Those are patients in the very early stage of the disease, anemia and blood transfusion dependency. First patient was dosed in May, and 2 additional patients were dosed in July and in September. Patient MNV014 compassionate use patient was dosed a week and a half ago, and this is actually the first patient to receive a second treatment of mitochondrial augmentation. She received a dose about 4 years ago, and that was with the maternal-derived mitochondria. So this is the first time that we are redosing patients under this program. Next, please. So this is still early. We can say that in terms of safety, there are no adverse events associated with the drug product, and we have a 6 months follow-up for the first patient that was dosed. And as you can see, in terms of the IPMDS score, it seems that the patient showing reduction in the total IPMDS score and in the complaints and symptoms. The lower the score is, the better the patient is doing. And there is also an increase in ATP production in his blood cells. We are still analyzing the 6 months follow-up time point, and in the next few months, we will have the other 2 patients coming up. Next slide, please. In terms of the anemia, so this patient had a low red blood cell count and a high MCV, as you can see, that after treatment, about 2 months after treatment, starts to get towards the normal minimal range that is acceptable. So in terms of the anemia, it seems that the patient is improving, but we do know that this parameter of anemia and blood transfusion dependency can also resolve spontaneously. If you look at the natural history of this patient, you see that he was deteriorating before the treatment, and then we observed an improvement. So that was encouraging, but again, still early to say. Next, please. For the longest follow-up, we have the KSS patient that was treated now 10 months ago. Up to the 6 months follow-up time, we again observed no adverse events and no anti-mitochondrial antibodies. This patient as well showed improvement in IPMDS score already at 3 months and at 6 months post-treatment, both in the functional, the physical, and the complaints and symptoms, and in the total IPMDS score. Her BMI that was deteriorating before treatment stabilized after treatment, and that was encouraging as well. And in the next slide, you can see her blood biomarkers. So you can see an improvement in ATP production in the PBMCs at 6 months follow-up time point, and a significant increase in healthy mitochondrial DNA copy number, meaning the full-length mitochondrial DNA in the blood. We will have the next time point follow-up in 2 months. And overall, again, this is encouraging. Next, please. What we have also done, and this is just a short update, because now we are headed towards discussions with the FDA. So we analyzed the 9 patients that were treated with our MNV-101. This is the first generation product for maternal blood. And we compare that to the CHOP published natural history led by Dr. Rebecca Ganeski from the CHOP. And what we observed is that the patients who were treated with mitochondrial augmentation showed a stabilization in the decline of height SDS, meaning these patients started growing post-treatment, and that was statistically significant, different from the cohort of the natural history. If you look at the middle graph, so you see that the patient at 6 months before treatment versus 6 months after, or at 1 year before treatment versus 1 year after, showed an increase in growth. And that's what we want to propose as a significant endpoint and an objective endpoint for the patients with Pearson. The patients also showed increase in, again, healthy full-length mitochondrial copy number, and this is the biomarker that we would like to propose in addition to the growth parameters. And in the next slide, you can see that actually the growth correlates very nicely with how the patient feels and functions. So the IPMDS score shows high correlation with the change in height SDS. The more the patients are growing, the better they are feeling. And this is the interactions that we are currently having with the FDA towards the design of a single pivotal study to approve mitochondrial augmentation for Pearson cases patients. And we are hoping for good news for the next update. So thank you very much for your attention.
Thank you so much, Natalie. Our next speaker is Dr. Robert Fisher. He's the CEO, CSO of Makos Therapeutics. Robert is a cardiologist by training with a research background in cardiovascular physiology. Today, he will provide an update on a Makos clinical candidate OMT28 currently being tested on primary mitochondrial disorders patient with myopathy and or cardiomyopathy in Europe. Welcome, Robert.
Thank you very much, Emil, and thank you very much, all your UMDF organizers. Next slide, please. That's just our general disclaimer, so maybe you go to next slide. Yeah, so Makos, for those who don't know us, Makos is a Berlin-based company. We develop a first-in-class series of molecules which are fully synthetic analogs of a specific metabolite from the omega-3 fatty acid EPA. Our molecules have anti-inflammatory and cell-protective properties, improve mitochondrial function, and hence we are interested in a variety of indications, but clinical-wise are clearly focused on mitochondrial disease and cardiovascular disease. Next slide, please. OMT28, our lead molecule, is targeting the interplay of mitochondrial function and inflammation. Next slide, please. Likely according to a recent publication, Nature Metabolism, via modulation of the sphingosine 1-phosphate receptor 1, which is downstream linked via activation of eNOS to activation of AMPK and SIRTWIN1 as known key regulators of mitochondrial function. Next slide, please. Interestingly, the sphingosine 1 pathway is not only relevant for cardiovascular disease and inflammatory diseases, but it is also a major role in the physiology of mitochondrial function. It has been linked to ATP production, mitochondrial biogenesis, ROS production, and mitochondrial calcium regulation. And this is all also supported by our own preclinical data for OMT28. Next slide, please. So the study we are currently running is our first study in mitochondrial disease. It's a phase 2A study in patients with mitochondrial tRNA point mutations, including M3243A2G, MA344A2G, and single mitochondrial DNA deletions. One specific feature of our study is that we select patients based on their baseline GDF15 levels. You might know GDF15 is supposed to be a surrogate marker of mitochondrial function. And we saw in a previous trial that patients with elevated GDF15 levels respond in particular to treatment with OMT28. The study is an open label study divided into 3 phases. We first all patients start with a natural history phase of 3 months, followed by a 6-month treatment phase, and then a follow-up phase for 4 weeks. Our primary endpoint, efficacy endpoint, is the responder rate in GDF15 in patients responding with GDF15 reduction. We also have a special focus on biomarkers, so we want to learn from that study on the relevance of biomarkers in mitochondrial disease, in particular in this population. And of course, we also included all the clinical functional readouts that you know, including 12-minute walk test, fatigue score, a 5-time citrus stand test, and others. Next slide, please. So the study is running in Europe, and all patients are now fully recruited. So thanks to our investigators here from these 9 centers, they really did a great job in enrolling patients for us. Next slide, please. Yeah, so I'm happy to share some first data on the study population. Since all patients are enrolled, we can share this part of the data. So as you see, in total, 29 patients were enrolled in a period of 12 months from these 9 centers, which I think is an excellent result. We have a somewhat equal distribution between male and female patients. Median age, somewhat typical, I think, for these types of studies, is with a median of 47. Mean 12-minute walk test distance is 694 meters, which is a bit more than in other studies. But we did not really select it for severe forms of myopathy. So that's likely the reason. As far as mutations are concerned, as you would expect, so the majority of patients carries the M3243A2G mutation, so nearly 70% of the population are carrying this mutation. Many patients have been diagnosed with cardiomyopathy, so approximately 50% of our study population, and almost everybody has a diagnosis of or clinical presentation of myopathy. Next slide, please. So with regard to GDF15, you see here the distribution. Most patients cluster in the range of 1,000 to 3,500 picogram per milliliter as expected. We only saw 10 patients which dropped out during the screening due to GDF15 levels either being above or below the set threshold. Next slide, please. So here you see a distribution of GDF15 at baseline, depending on gender and age. Females tend to have a bit lower GDF15 levels, which might be just due to the smaller body or body muscle mass. Otherwise, you see here also the distribution for age. And clearly, there's no dependency on age, which you would expect for the general population. So which means that these elevated GDF15 levels are really due to the underlying mitochondrial disease. Next slide, please. And somewhat the same is true also for these different mutations. For some of the mutations, we of course only have low patient numbers, so this is very limited conclusion. But clearly, there seems to be not a real dependency, at least for these mutations here in our study for GDF15. Next slide, please. With regard to comorbidities, as everybody would expect, so our patients have many manifestations for different organs headed by endocrine or metabolic diseases. So many of our patients have diabetes or hypothyroidism, but they also have diseases of the nervous system, ear diseases, and diseases of the circulatory system, as well, of course, diseases of the musculoskeletal system. I think that's a clearly typical contribution of comorbidities in our study. Next slide, please. We, of course, one specific focus is also cardiomyopathy in our study. I think this is somewhat different to other studies that have been done in the past. So we included measurements of echocardiography for different time points during the study. And one first finding is maybe that we see more of left ventricular hypertrophy than expected from the medical history. That's maybe, probably many of you would say that's also not unexpected. And with regard to left ventricular ejection fraction, most of the patients have normal ejection fractions, at least in our study. 5 patients had slightly impairment of left ventricular function, and only 1 patient has really significant impairment below an ejection fraction of 40%. Next slide, please. So this is my last slide. So as I said, so all patients have been enrolled in this study. The safety and tolerability of our molecule is confirmed so far in the data. We have some first clinical data, which is looking promising, and expect last patient out in May 2025. So then please stay tuned for the results then in next year. So thank you very much to everybody.
Thank you, Robert. That was very interesting, especially with the GDF15 data. I would like to remind everyone that we're not doing any live question and answer, but if you have any burning question, you can always put them in the chat and the UMDF staff can reach out to you if there are any answers available. Our next speaker is Patrice Rieu. He's the CEO of Thiogenesis. Patrice is the co-founder and director and chief executive officer of Thiogenesis Therapeutics. He has been deeply involved in the development of drugs for rare diseases over the last 20 years. And today, Patrice will introduce Thiogenesis' clinical development plan for Leigh syndrome. Welcome,
Okay. Can you hear me?
We can.
Okay, good. Thank you very much, everybody. So yeah, thiogenesis, you might know the name now, I hope. And next slide, there is a standard one for the finance guy use generally. So let's talk a little bit about Lee syndrome. Everybody knows about Lee syndrome now. They use the term Lee syndrome system or something like that. So I don't want to go into the clinical, but basically, from a biological point of view, the patients with LSS, they have actually a very decreased depletion of glutathione and an increase in the disulfide, which is the GSSG, which is a specific mark of oxidative stress. Oxidative stress means a lot of ROS, increased production of ROS. And what happened is that the mitochondria is effectively the main source of ROS. Next slide, please. And the issue with the mitochondria and ROS is that very few compounds go into the mitochondria and to neutralize this ROS. And that's why all the compounds that have been developed before by MitoQ or even AP743 or all this quinone, and they don't go very well into the mitochondria. Only two antioxidants go there to neutralize the ROS. It's actually glutathione and thioredoxin, because there is a specific transporter for each one. Thioredoxin is a protein, it's difficult to use, you have to inject, and it's not really used for that. So the only one possible is really the glutathione. Glutathione goes into the mitochondria through a specific transporter, and when there is more ROS produced in the mitochondria generate more, there is upregulation of the transporter. So there is more transporter for glutathione. But the issue is that glutathione has to be transported in the mitochondria because it's not produced in the mitochondria. And when there is more transporter, there could be more glutathione transported into the mitochondria, except that if there is not enough glutathione outside in the cytoplasm, there is no more glutathione in the mitochondria because the transporter itself doesn't signal to the cell that there is a need for more glutathione. So producing more glutathione is relatively easy. It's not, you cannot eat it orally or inject, or it's also it's very quickly destroyed. So the only way is to produce inside the cell the glutathione. That's easy if you have glutamate and glycine, which is easy get. That's a compound that are very well available, but there is also a need for cysteine. Cysteine, which is the third compound to build glutathione, is very well regulated in the cell. So the level of cysteine is, as soon as it increases, it's actually very well balanced quickly. So the only way is to have a different way of increasing cysteine instead of just providing cysteine. And to do that, the only good way, actually, or probably the only good way, is to get the cysteine where it's stored. And it's stored in the lysosome, which is a trash can for the cell, basically, and it's stored in terms of a disulfide. Being stored in terms of disulfide, you have to break the disulfide, which is cysteine on the left here, into 2-cysteine. And you do that, we know that there is a good way to do that, which is when patients have cystinosis, they have a problem to transport the cysteine outside. So we know that if with cysteamine, we can break easily the cysteine into 2-cysteine and break that. So we have a production, we can generate more cysteine that goes into the cell. And then when there is more cysteine into the cell, we can get more glutathione into the mitochondria. The only question at the moment is the quantity of cysteamine we need. We know that for cysteine, there is a lot of cysteine in there, but in normal cells, there is a little bit less, but we don't. That's the only question we have on the concern here. So next slide. We presented a strategy to the FDA with a group from a big clinical site in the US, and having two phases. Phase 2A, which is a study where it's open label with 10 patients, and the we think 10 patients, that's a discussion. And then looking at parameters that could be useful for the development, that would be useful for the development of the next trial. The next trial being a phase 2b/3, where we could, after that, register the drug for marketing. So the idea, when we submitted that to the FDA, they say, oh, okay, that's actually good. You could do even 3 adults, starting with 3 adults and then starting with kids. But we really want to help you, which means that we were expecting to start very quickly, but they want to help us in the sense that they want to make sure, and that makes sense. They want to make sure that we are using especially the right primary endpoints to help us design the next trial, the phase 2b/3, with the right parameters to determine that effectively it's working, and especially because if we are dealing with children very quickly, make sure that there is really a benefit for as soon as we can show it for the kids enrolled in the study. So that's where we are. We are resubmitting very soon a very more detailed protocol with helped with UMDF actually to determine all these parameters and figure out the best design for first getting some idea about the efficacy on increasing GSH and improving efficacy clinically for the patient, but then having a way of comparing versus placebo the next phase 2b/3, which should be next year, actually at the latest, to get the drug approved for Lee syndrome. In parallel, next slide, we have been working with for developing because it's almost the same kind of pathological or biological issue with a generation of ROS. And so that's MILAS. We are working on MILAS in Europe. In Europe, we submitted an IMPD. We got approved in July for the core of the protocol, the IRB and everything. It's actually done. And they say, now you have to go for the part 2. That is, make sure that for each country, you have all the budget and the ethical committee and everything like that. So next slide. So that's 12 patients we're going to enroll with placebo versus placebo, 8 under treatment and 4 placebo. We'll do an interim analysis. There are 2 sites, one in France with Dr. Magali Bart and actually Vincent Procaccio, and another one with in the Netherlands with Dr. Janssen. So we have all the paperwork done for the Netherlands. It's done in parallel, and we have almost everything done for France. But for example, and that's where we are also delayed. We were expecting to start before the end of the year, but you know, last 2 weeks ago, the French actually agency said, okay, we reviewed the protocol, and you know, in the protocol you have inclusion criteria and exclusion criteria. We'd rather have inclusion criteria and non-inclusion criteria, and so we submit the modification, and they say, okay, now we'll tell you at the latest in 30 days what we think. So we are still waiting and waiting. That's paperwork, but that's okay. So that's where we hope that beginning of next year, we will have both clinical trials starting in Lee syndrome in the US and MILAS in Europe. Thank you.
Thank you very much, Patrice. Our next speaker is Dr. Vijay Mudur. He is the CMO of PBG Bio. Dr. Mudur oversees PBG Bio clinical development efforts with the company's portfolio of transformative medicines based on a deep understanding of biological pathways derived from the NAE platform. Today, he will provide an update on the company's clinical plans to address primary CoQ10 deficiency.
Thank you very much for this opportunity. We want to present our plans for going into clinical trials in primary CoQ10 deficiency. Next slide. What is primary CoQ10 deficiency? It's a rare, debilitating mitochondrial disorder which affects multiple organ systems and is caused by mutation in one of 10 genes involved in the biosynthesis of CoQ10. The deficiency may be mild or severe, and when it is severe, it can lead to early mortality. In the severe cases, like every mitochondrial disorder, it can be very severe encephalomyopathy, ataxia, multi-system disease, and sometimes it can present as isolated myopathy, though that's rare. The advent of next-gen sequencing has really changed how these patients are diagnosed. They're showing up earlier and earlier. Sometimes they're diagnosed with whole exome sequencing in the NICUs. It is considered a treatable disorder. Patients get CoQ10 over the counter orally. Next slide. However, the over-the-counter oral CoQ10 therapy seems to have low response to therapy. And this has been published in numerous occasions. There's one study which Wang and Hikami conducted where they reviewed the literature. And in the literature, they logged case by case what the response rate was. And out of 89 qualified patients in the literature who they could get, only 22% demonstrated an objective response to over-the-counter oral CoQ10 treatment. Now, lack of response can be due to many factors, but one of the major factors is about the molecule CoQ10 itself. As Patrice had previously presented, very, very few things get into the mitochondria. In fact, with CoQ10, it is so insoluble, very little of the oral CoQ10 gets into the circulation, and even less gets into the mitochondria. So how do we fix this? In the next slide. BPM 31510, it's a nanotechnology-based formulation that penetrates the mitochondria. We have used BPM 31510 in multiple models in oncology, and we have shown that the drug conjugate increases the mitochondrial Q pool. And in oncology, we've seen, especially in many, many models, we've seen durable therapeutic benefit. This formulation results in differentiated bioactivity and bioavailability. It is also well tolerated. What does it do in cells from patients with primary CoQ10 deficiency? To ask this question, we worked with Dr. Quincy at Columbia University, where healthy fibroblasts and CoQ2 mutation fibroblasts were taken, and they were cultured in galactose medium. The reason galactose is used is when fibroblasts are cultured in galactose medium, they are forced to use the mitochondria to survive. And what happens is healthy fibroblasts do fine irrespective of the galactose medium, irrespective of whether they got CoQ10 or didn't get CoQ10. However, fibroblasts with CoQ2 mutation, when CoQ10 was added, you would expect them to survive. But with this severe stress on the cells, growing them in galactose medium, they didn't survive. But the same amount of CoQ10 packaged in BPM 31510, all the cells survive. And we know this is not because the CoQ10 isn't reaching the cells, because even when you measure CoQ10 in the CoQ2 mutant fibroblasts, they do have a higher than normal level. So this really got us thinking. Next slide. We also asked, is it just going into the cell, or can it get into the body better? So we took mice, we gave them oral CoQ10, or we gave them BPM 31510 at the same doses. And what we know from our clinical trials in oncology, we get a very high level of plasma CoQ10 when we give it as BPM 31510. And then we found out in our studies, we get a high muscle level, high heart level, high kidney levels, and we can also get into the brain. And you can see a kind of a ladder effect. Some tissues, a lot of it goes, some tissues, less of it goes. Next slide. We also look more deeply. Is it enough to get more CoQ10 in the blood? So here, what we did is we did a technique called mass spectrometry that can measure molecules on slides that are similar to histopathological slides. Here we took mice that were treated with vehicle or BPM 31510 and looked at CoQ10 levels and other quinone levels and asked if it is going into the mitochondria by tagging the cardiolipin. Cardiolipin is a molecule found only in the mitochondria. And what we see is BPM 31510 results in very high levels of CoQ10 and other quinones in the mitochondria in vivo in the muscle, showing that not only does it work in cell culture, not only does a lot of it get into the circulation, it also gets into the cell and into the mitochondria. Next slide. We have clinical trial experience with BPM 31510 in oncology. In trials, over 200 patients have been treated. And in oncology trials, it's frequently in combination with chemotherapy, but we've treated 95 patients with BPM 31510 alone. Patients have received as greater than 400 milligram per kilogram per week of BPM 31510. And what we expect is when we start doing trials in mitochondrial disease, the dose of BPM 31510 may be 10 times lower or potentially even lower than the highest doses we have given in oncology patients. As is known with patients with mitochondrial disease, oncology patients also have significant intercurrent illnesses, and even in those patients, it was well tolerated. And it actually works well with chemotherapy, which is not relevant for this current situation, but is important to know because a lot of chemotherapy are also mitochondrial active. Next slide. So what we have done is we spoke with numerous mitochondrial disease experts, and there were requests made to make BPM 31510 to become available on a named patient basis. We have approved treatment for 3 named patients. 2 patients have already been dosed. Over 25 patients have been brought to BPG Bio's attention as potentially having poor response to oral over-the-counter therapy. We are considering doing a trial, and we are planning a clinical trial in primary CoQ10 deficiency. What we plan to do is, with these requests for named patient use, we will get data from 4 or 5 compassionate named patient treatment, which would guide the design of our trial. Next slide. We have also been alerted to the possibility that BPM 31510 may have potential utility in other mitochondrial and related diseases. CoQ10 is a key component of the mitococktail, and there's anecdotal evidence for treatment benefit with CoQ10 in patients with certain types of mitochondrial and related diseases. So what we are doing is we are working with numerous experts in the field. We welcome others to come and talk to us. We would love to generate the scientific evidence for treating other mitochondrial diseases and put together a program where we can give BPM 31510 across many mitochondrial diseases where patients may benefit from therapy based on robust evidence generated through rigorous research. Thank you very much. That's the end of my talk.
Thank you, Vijay. Our next speaker is Sharon Ham from Sail Therapeutics. Sharon is one of the original co-founders of Sail in 2015, and she continues to lead the technical operations team with the responsibilities for all drug development and regulatory handovers. Today, she will provide an update on Sail's clinical development program focused on pyruvate dehydrogenase complex deficiency. Welcome.
Thank you very much. Appreciate the opportunity to be here today. Next slide. On behalf of Sail Therapeutics and our development partner, Dr. Peter Stackpool at the University of Florida, we're particularly pleased to be able to give an update now on the status of activities that we've been involved in that connect to the PDCD community at large. These would include that we've been involved in a awareness, of course, of the update that's been given to FDA from an FDA listening session where families shared their experience with FDA. We've been involved in an expanded epidemiologic assessment of PDCD to try to better understand what that epidemiologic background looks like in this condition. We've now completed our 2 phase 3 trials and submitted our NDA and are still supporting emergency IND requests while we expand our expanded access program opportunity for other patients. And we'll talk a little bit about that in the slides to follow. Next. From an epidemiologic perspective, I would say that the big learning for us is that while published data would suggest a number of patients that are have PDCD, our learnings here would suggest that, in fact, the number is probably bigger than was originally thought. And part of that is because roughly 50% of the patients are currently not diagnosed with PDCD. And so the numbers could be as large as 2,000 patients in total. Many of these patients are undiagnosed until they are after the age of 1. And that's despite the fact that the majority of these undiagnosed patients are often seen at some point by a pediatric neurologist. Next. As I mentioned earlier, we've had 2 phase 3 trials that have just been completed. The first of those trials was a phase 3 trial where we had a double-blind placebo-controlled trial using the treatment of dichloroacetate and that versus a placebo. There were two active treatments. You were either on an active treatment crossed over to placebo, and then, or the reverse is true. You started on placebo, crossed over to an active treatment. We had 34 patients who completed the study itself, and those patients were assessed with a primary endpoint for a change in their observer-rated outcome and a secondary endpoint that looked at lactate in particular as a biomarker. All those patients then continued onward for an open label extension program, and many of those patients are still actively involved in the open label extension. Next. In addition to the phase 3 trial that I mentioned before, we have a second study that actually also assessed the opportunity to look at the natural history of the disease and how that compared to the patients who were treated in our phase 3 program. And in that context, this was an overall survival analysis where we looked at the underlying survival with a mortality rate published by DeBras at 39%, and how that compared with patients that were then on active treatment during the course of our program. Next. In addition to running the programs that I mentioned here, we've obviously expanded that now to look at patients who didn't have an opportunity to participate in trials. And so on the one hand, I'd say first thank all those patients and families who did participate in trials and the investigational teams. But we're also looking now to have an opportunity for patients who are interested in moving into treatment with DCA as we open up an expanded access program. This program will be open for all patients of any age with a confirmed diagnosis of PDCD. You would undertake a GSTZ1 haplotype test first so that we could understand your ability to metabolize dichloroacetate, and then you would be randomized into one of two groups, whether you're a slow or fast metabolizer with respect to the dose of the product. We are using a specialty pharmacy for this effort, Novo, and they will be involved in handling program administration. And there will be a lot more information on this as we kind of approach 2025 moving forward. Next. As we look forward, it's a bright future ahead. Obviously, we're excited about being where we are from a development perspective. We're in the process now of having just filed the NDA, so we're working with FDA on that NDA submission and its review here as we move into 2025. We'll be creating the infrastructure that we're going to be needing to support the expanded access program that will be opening in 2025 as well. And we're looking to continue to understand the diagnostic journey and the disease awareness aspects for PDCD and what we can do to be helpful in that understanding as well as we advance to commercial launch. And for that, I thank everyone for their time and attention.
Thank you so much. Our next speaker is Susan Van Meter from UCB. Dr. Van Meter is the physician leading the MT1621 development program, and she has been in industry for almost 20 years with a focus on rare disease drug development. Welcome, Susan.
Thank you, Amel, and thank you, UMDF, for the opportunity to give you an update on the doxycytine and doxorubicin program, which is formally called MT1621. Next slide, please. This is just a disclosure slide. Next slide, please. Thymidine kinase 2 deficiency, or TK2D, is an ultra-rare progressive disease that is often life-threatening. It's caused by an autosomal recessive mutation in the TK2 gene, which is crucial for the maintenance of mitochondrial DNA and ultimately energy production. It is mainly associated with proximal muscle weakness or myopathy and respiratory weakness, and has a high mortality rate. Many people lose the ability to walk, to eat, and to breathe independently. It can be misdiagnosed for other neuromuscular and mitochondrial myopathies. It affects almost every aspect of the patient's life and is very debilitating, and there are currently no approved treatments. The current management consists of supportive care that involves many physicians and other healthcare providers. Next slide. Doxycytine and doxorubicin is a combination of two drug substances being developed as a treatment for TK2D. Doxycytine, or deoxycytidine, is one of the components, and doxorubicin, or deoxythymidine, is the other component. Next slide, please. So we're using preliminary evidence and leveraging a lot of information to go to regulatory submissions. Fortunately, the pathophysiology of TK2D is well known and well characterized, and we are able in our non-clinical program to confirm target engagement with an increase in mitochondrial DNA copy numbers across most of the tested tissues, including skeletal muscle, with associated changes in respiratory chain complexes that ultimately lead to ATP or energy production. We use real-world evidence data from untreated patients to confirm the natural history of the disease, and as an external control cohort to understand survival benefit. For the treated patients, we're building on initial findings from the first data review study, MT 1621-101, to evaluate survival benefit and other changes in disease trajectory following treatment. We're using clinical data from an ongoing open label study, TK0102, as well as data review studies, MT 1621-101, MT 1621-107, and company-supported expanded access programs. We're using all available regulatory pathways to ensure alignment with the European Medicines Agency and the FDA. In the US, we have what's called breakthrough therapy, which has allowed increased communications throughout the development program. And in Europe, what's called the PRIME designation to work with EMA on developing the pathway to demonstrate the substantial evidence needed for benefit and safety of doxycytine and doxycytine. Next slide, please. So where we are now, we are continuing to develop doxycytine and doxycytine as a treatment for pediatric and adult patients with TK2D with an age of symptom onset on or before 12 years. We have an active compassionate use program with company-supported requests from treated physicians evaluated on an individual basis. And this year, we have also initiated a company-sponsored compassionate use or managed access study. We support the mitochondrial disease community with disease education. And as noted below, we are preparing for marketing authorizations in this year. So look for more information on this last item early next year. And that concludes my program. Thank you.
Thank you, Susan. Our last speaker of the day is Katie Lance. She's the Executive Director at Stealth Biotherapeutics and serves as the Head of Patient Advocacy and Government Affairs. She is responsible for building and maintaining relationships with all patient advocacy organizations in the mitochondrial space and prioritizes elevating the patient caregiver's voice in all aspects of drug development and beyond. Katie, welcome to our community, and please take it over.
Thank you so much, and thank you, UMDF, for hosting this webinar today. Next slide, please. Just recognizing time, I'll give a very brief overview of Stealth Biotherapeutics. We are focused on treating serious diseases of mitochondrial dysfunction. These range from ultra-rare diseases like Barth syndrome to more prevalent diseases like dry age-related macular degeneration. For purposes of today, we'll be focusing on an update around our Barth syndrome and primary mitochondrial myopathy programs. Next slide. For background, I wanted to provide a little bit of an update on the Barth syndrome development program. This program started back in 2014 when the Barth Syndrome Foundation approached Stealth regarding development of elomeprotide for the treatment of Barth syndrome. And in terms of trial design, the TaskPower trial was a double-blind placebo-controlled crossover trial with a 3-month treatment period that included 9 subjects. After that, we went into the TaskPower extension trial, which was an open label extension that lasted up to 3.2 years. I also wanted to highlight the natural history control. This is something some of my other colleagues spoke to earlier, but for this audience, it's really important to understand that the participation in things like the MitoShare registry enable companies to utilize that for regulatory review purposes. So Because the Barth Syndrome Foundation had a patient registry, we were able to use that as a natural history control. And finally, we have an active and ongoing expanded access program. Next slide, please. In terms of regulatory updates on Barth syndrome, I did just want to level set with a little bit of background on Barth syndrome. It's an ultra-rare, progressive, debilitating mitochondrial disorder that impacts around 130 Americans. In terms of symptomology, we have skeletal myopathy, progressive and evolving cardiomyopathy, neutropenia, growth abnormalities, and feeding problems. There's an extremely high mortality rate with 85% of deaths occurring before the age of 5. The TAS-POWER trial was the first and largest clinical trial effort in the Barth syndrome space. Our new drug application, or NDA, was filed this year and accepted by the FDA. It is currently under FDA review. Back in October, our program had a meeting of the Cardiovascular and Renal Drugs Advisory Committee, so an advisory committee meeting. Thank you to Phil for coming in person to show support, and thank you to the UMDF Patient Advisory Council for their submission on the public docket as well. I won't play this video clip here, but hopefully when the slides are distributed, you can watch the public link of the YouTube here. And I just wanted to highlight this as an opportunity that demonstrates really the importance of the patient and clinician testimony. As you'll hear, if you do have a chance to watch this, patient and clinician testimony, the KOL testimony, was critical to the understanding of the disease severity for the members of the advisory council. Ultimately, that advisory committee voted 10-6, concluding that elamiprotide is effective for treatment of patients with Barth syndrome. Our PDUFA date is January 29th of 2025, so early next year. Next slide. Sorry, and it's probably going to play. And just wanted to write a little bit more about our proposed indication for elamiprotide. So it's a daily subcutaneous injection, and it has a very well characterized safety profile with over 400 years of patient exposure. Next slide. In regards to our primary mitochondrial myopathy program, just wanted to highlight the importance of learning from clinical trials. So we had the largest phase 3 mitochondrial myopathy trial, the POWER trial. And based on some of the data that we saw there, given the disease heterogeneity, we decided to do another phase 3 mitochondrial myopathy trial, this time focusing on those with nuclear DNA mutations. So if we can just go into the next slide, please. So as I said, some of the learnings that we had as we went into the phase 3 new POWER trial was around the myopathic phenotype, increasing the dosage. So we were originally at 40 mg and we moved up to 60 mg, and then a longer duration. And this is how learning from other trials can really help inform trial design. So with our Barth syndrome program, we started to see that some of the peak results were around 9 months of therapy, and that helped inform the new POWER trial to increase the duration. Next slide, please. Just to provide a view of the trial design here, it's a double-blind, placebo-controlled trial, a 48-week in terms of length, and we have about 90 patients enrolled. The primary endpoint is changes from baseline for 6-minute walk test. Secondary endpoints include 5 times sit to stand, 3 times up and go, and patient reported outcomes with a global severity scale. This is fully enrolled, and we are expecting top-line results early next year. So 2025 will be an exciting year for Stealth Biotherapeutics with both our Barth syndrome PDUFA date early next year, as well as top-line results here. I think that put us right on time, so I will conclude my presentation. Thank you all.
That is perfect. Thank you for staying on time. This concludes our update session today. Thank you to all the speakers. And Phil should be joining me today for a concluding remark.
I'm here. Yeah, that was really great. Amel, I think you'd join me in first thanking all the speakers, but also just sharing this really hopeful message with our patient community around the level of clinical research and clinical development that's taking place. We heard examples of drugs that are very close to being reviewed by the FDA and could be approved in the near future. We heard currently active clinical trials and even some trials that are still in development. And I think both you and I know there's a lot more behind that preclinical. It's a really exciting time for the community.
Yes. And we didn't have the chance to hear from some companies that are still running ongoing clinical trial in the US that will come to at a later date. But it's very exciting to be living this era where we have so much interest and so many opportunities out there.
For sure, definitely more there. And that's actually a perfect segue to highlighting what's going to be coming in January of 2025. We will continue with the clinical trial update theme. We will have several companies presenting: Condrion, Tesento, Abliva. All of these all of these companies have clinical developments that involve BLAS 3243 mutation. So we're going to combine that together with Fernanda Scalia from Baylor, and also have a presentation by Ingrid Tyne from Hospital for Sick Children in Toronto. So it should be a really great update in that specific area of mitochondrial disease. And we hope all of you will join us on Monday, January 13th, 2025 at 12 o'clock for that webinar. So Amel, thank you for your moderation. With that, I'll wrap up. Wish everyone a very happy holiday season and see you in 2025.
Happy holiday, everyone. Bye.